Thank you very much for hosting us. We're delighted to be here. My name's Sam Barker. I'm Chief Business Officer at ViaNautis Bio. And we're a genetic medicines company based here in Cambridge. Brilliant. So I think I assume everyone in the room is familiar with genetic medicines and the advances that it's made over the past 10 years. And a lot of this has been driven, I think, by disease biology and payload design and the way in which they've advanced. But what it leaves as a bottleneck is delivery. And this is constraining genetic medicines from being able to move away from niche, rare, severely life-threatening diseases into broad patient access and large indications. And that's where we come into the picture. So we've developed a technology called polyNaut. It's a single polymer nanovesicle for targeted delivery of gene therapy. And what we'll talk through today is some of the advances we've made in our platform and then how we're building that into our pipeline. So let's talk a little bit more about the problem. The first generations of genetic medicine have really been enabled by delivery using viral vectors or lipid nanoparticles. And there are some significant structural constraints that are maintained with these delivery methods, despite many billions of dollars and years of development. So viral vectors still struggle with scalability of manufacture, but crucially safety and redosability. And the first generation of lipid nanoparticles has enabled a massive industry for vaccine delivery, where mRNA has been enabled for broad delivery without the need for specific targeting or necessarily frequent redosing. As we've seen targeted generations of lipid nanoparticles come in, that's built in significant complexity of manufacturing, and we still have a question over the safety and tolerability of these delivery particles, especially when we want to move into the therapeutic application of mRNA as a protein replacement. So polyNaut comes into this. It's a single polymer nanovesicle, and we've been able to demonstrate really high quality product attributes that means this will be able to enable new genetic medicines into much larger patient populations. So in terms of quality attributes, we see significant stability at 4 degrees without the need for -80 cold chain supply. Manufacturing, I'll talk a little bit more about later. We've really focused on the scalability of our manufacturing and de-risking that. And then just looking at the IP space, this has become incredibly crowded for viral vectors and targeted lipid nanoparticles and polymer IPs. It's completely clear of that. So we have strong protection, as well as the ability to work with multiple different cargo types, which will exemplify with great tolerability and safety predicted. And lastly, and one of the most key differentiators of our platform, we have efficient targeted delivery and modular plug-and-play mechanism. So I won't spend too much time talking about differentiation, but what we have seen is that as targeted nanoparticles, particularly lipid nanoparticles, have been developed, we're bringing in multiple different components. And this really increases the complexity and the manufacturability of these TLMPs. And what we haven't seen yet is how that translates into challenges of safety and scalable therapeutic delivery. So we need to see from these targeted lipid nanoparticles that they can deliver multiple doses. We haven't seen that yet, but what we do see is the cost and complexity rising. On the other hand, if we look at our polyNaut nanovesicles, the benefit of using a polymer is that we have a single polymer component. It's a biocompatible copolymer that self-assembles into a nanovesicle with an optional click chemistry reactive group for adding the targeting. And over the past 12 to 18 months, what we've been able to translate through is going from bench scale manufacturing into a fully automated CDMO compliant process. And as I mentioned, this is covered by our own IP, which is separate in a much broader open space than in lipid nanoparticles. So to focus in on manufacturing, this has been a key challenge for most delivery platform companies. And delivery platforms fall short when you can't control formulation. So we focus very much on moving into GMP-compatible automated microfluidic systems. And so we have well-characterized single polymers going in along with our cargo, which is an mRNA in this case, and that leads to a well-defined and quantifiable product with clear attributes. Beyond this, we now have modular post-assembly nanobody functionalization. So when we wish to create new PNVs, all we need to do is change the nanobody that we conjugate. We don't need to reformulate the whole PNV. And then lastly, as I say, we're now working to build this into a CDMO-compliant GMP manufacturing suite. Focusing in a bit more on the targeting, this is again where other delivery platforms are not able to meet the demands to enter into significant patient populations. So what we've demonstrated here is we're able to build a modular system whereby we can stably conjugate a modified nanobody with targeting specific cell populations. And shown here is an example of that, where we're targeting specific immune cell types. So we have very highly specific targeting, and we don't see tropism towards the liver or lung or any other peripheral organs, which supports the specific delivery, but also a much higher safety profile. So moving on from description of the technology, we've got a modular platform where we can target multiple different cell types and we can deliver a broad range of cargoes. We're now looking into how does our pipeline look? So what does that enable us to do? So we're active building our own wholly owned pipeline in the CNS, in cystic fibrosis, and also in I&I and oncology. In the CNS, we've been able to show that we can cross the blood-brain barrier and deliver genetic cargoes to the brain. And this unlocks large indications in the CNS where safe delivery and repeat dosing is critical. The cystic fibrosis program is supported by the CF Foundation, and here this is an inhaled delivery of full-length CFTR. So this is, again, targeted delivery, but it's opening up a patient base, not only of patients who aren't eligible for CFTR modulators, but also as a support for those who are responsive. So both of these programs have benefited from early validation through partnership. So we have an early stage partnership with the Cystic Fibrosis Foundation, despite the CF program being a wholly owned program. And we also signed a research and collaboration license agreement with Eli Lilly in 2024. And these both give us commercial support, but also have helped us to develop the platform and really push it to its limits and shape the areas that we're working into. The third program in our pipeline is the most recent addition to our portfolio, but perhaps the most accelerated. And this is really looking at targeting immune cells for development of an in vivo CAR. In this space, we're highly differentiated from lipid nanoparticles. And what we're looking at developing here is an in vivo CAR, building not only into I&I and oncology, but also looking at a safety profile for autoimmune diseases. And in terms of partnering, we're both building our own pipeline, but we also work with pharma in a way that we can have multiple different structures that work for both parties. So this can either be a platform-based collaboration, it could be around targeting delivery, or it can even be a product-level partnership. And then just to quickly focus in on our I&I program. So ex vivo CAR Ts have really demonstrated that this is an amazing therapeutic opportunity, but they're still significantly limited in their commercial success by the manufacturing challenges. And so in vivo CAR has really grown in the past year, and we've seen significant deal interest in this space, moving from lentiviral delivery towards non-viral delivery. And the point at which we're seeing significant deals is even moving into the preclinical space because of the potential shown here. So we're developing a PNV-delivered mRNA for developing an in vivo CAR. And here we really benefit from independent IP, using a single polymer formulation that has high stability, so great product attributes, meaning that we have the potential to move out of the hospital setting and into the community-based setting, which is essential for patient access and expanding that market. And as well as that, having specific or highly exquisite targeting towards specific T cells, and then the potential for redosing, so being able to supplement that response, which we've seen already exemplified with ex vivo CAR. So this is a really exciting program for us, which is going to be developing through the next year. So just lastly, I want to kind of wrap up and answer the question, why would you want to invest in ViaNautis? So we've put a huge amount of effort over the past 2 years with the support of our current investors to exemplify the platform and validate our platform, and now build into our pipeline and our programs. And through the next year, we're going to see some really significant value inflection points as those programs generate preclinical data towards development candidates. We have a very strong team. We're at around 40 people now, based in the Unity campus, and a strong record of company building. The platform is very well validated, and we're working in what I think are really exciting and meaningful therapeutic areas. We have a very strong IP portfolio that's separate from LNPs and AAV and gives us a potential for 20 years protection, and multiple ways in which we can create value through our own pipeline, but also through BD, partnering with pharma, and through bringing in new products as we develop the platform and as we go along. We're validated by our global partners, so we have excellent early stage partnerships and also highly credible investors. So through 2026, we look forward to bringing in additional funding, which will help us take our programs even further on, and ultimately meet our aim of taking genetic medicines to a much wider patient base in currently underserved diseases. So thank you very much for your attention.
Question also. Have you done side-by-side comparison with LNP to see how much better your technology is versus alternative technologies?
Yes, certainly. So we've developed comparison LNPs, that what we would consider to be the leading targeted LNPs, and we're running certain studies like biodistribution and safety and efficacy alongside its head with what we would consider to be benchmarks.
So you're waiting to see what the differences are.
Yeah, I think that's something that will come through this year.
Being a typical investor, we've gone in with conviction, but we've hedged our bets. We've got another LNP company. No, they're good. We incubated Camgene evidence for the seed them doing very well. But yeah.
How much you raising?
So we haven't yet launched our Series B, but we'll be looking at doing that through the next year. And we would like to raise enough that we can take at least one of our programs into the clinic through to an inflection point. So we will see if there's interest to start with one program, it may be that we're expected to.
And the IP for the program was that?
We build up the IP, so we have a composition of matter IP around the components, and then it builds into both platform, and as we create a PNV that's got both targeting and cargo, that creates product-based IP. So we try to layer that, and that also supports both our protection but also our partnering.
Great, thank you.
Good.
Cheers. Everyone had the chance.